morning, happy Friday, and welcome to the Friday PowerTalks series. I'm very happy to introduce today's speaker. It's Dr. Christopher Willey Md, Ph.D. He's Hale Stevens War Professor in the Department of Radiation Oncology. He's also Director of the UAB Kinomics Corps, and he's well known to us. He's been very much involved in a lot of our informatics activities. He also has appointments in chemistry and molecular genetics, academic joint departments, cell developmental and integrative biology, and biomedical engineering. And also he's scientist appointments in multiple institutes. His focus is in cancer biology, and he's a radiation oncologist. He's interested in the response to radiation, as well as abnormal signaling that governs cancer biology at the individual tumor level, or even the cell level, and developing treatment strategies based on that. Also focused, and he's presented previously on kinases, which are proteins that control signaling, and is very much involved with cancer development, and a focus on glioblastoma multiforme, which is brain cancer, one of the most difficult to treat and the most aggressive. Did his bachelor in science and engineering at Duke, and his MD in University of South Carolina, as well as PhD in molecular cellular biology and pathobiology. So I'm very happy to introduce him, and his topic is multi-omic translational approaches to investigate therapeutic resistance in glioblastoma. So take it away.
All right, thank you for the kind introduction. So yes, I definitely have been meeting with folks in the Informatics Institute and bioinformaticists for several years now, as research continues to increase in terms of the amount of big data utilized and even how we're utilizing it in the clinic. So this is kind of going to be a whirlwind tour on kind of translational work related to resistance to therapy in glioblastoma. And so certainly can't cover everything, and certainly it's going to feel like more like a 10,000 foot view for a good portion of it. But I am going to focus a bit more on some of the work that we're doing, particularly with Jake Chen's group and Laura Einoff over in Civitan. So just a couple disclosures regarding funding and some of the things I do, not really directly related to this presentation today, though. All right, so glioblastoma, also called GBM, is a really bad disease. So I know, at least for this audience, some of this may not be that familiar to you. This is a sagittal MRI showing one of these lesions inside the brain of a patient. It's a pretty devastating disease. We do, we treat it, here at UAB, actually a decent number. It's the most common primary malignancy of the brain, so meaning a tumor that originates in the brain. The most common brain cancer is actually metastasis from other sites, but GBM is particularly difficult because of its aggressive nature and resistance to therapy. You can never cut it out because it's highly invasive. And so, we really have struggled to make a big dent in terms of improving survival compared to, let's say, other cancers out there. Currently, the standard of care is maximal safe surgical resection, which may be as little as a biopsy, or maybe what we would call a gross total resection, where at least the enhancing portion of the tumor is removed. And then we add on a chemotherapy called temozolomide with radiation, and then afterwards they get additional chemo and potentially tumor treating fields, which is an electronic device. So how did we really get to that point? Well, the landmark trial was 17 years ago. Dr. Stoop and his group basically compared the standard, which was surgery and radiation, with the addition of temozolomide. This is kind of an interesting alkylating chemotherapy that actually can penetrate into the brain well, and it's oral. So it's just a pill they take nightly. And it turns out that there actually was a pretty nice bump in the survival curves, as you can see here, on the overall survival and the progression-free survival. What you'll notice is that, we are talking about months here. So you still have a very small portion of patients alive at 3 years with this kind of new standard. And so a companion paper kind of started to delve into some of the really biomarkers and both predictive and prognostic. And so in the same issue in New England Journal, they published on an epigenetic silencing agent, basically MGMT, which is this 0,6-methylguanine DNA methyltransferase. Basically, it's the antidote to temozolomide. It can remove the methyl group that temozolomide adds to DNA. And so when patients have the MGMT promoter silenced with methylation, then they really can't repair the temozolomide injury. And so those patients live longer and potentially even respond better with the combination therapy. And so this was kind of the beginning of really starting to put predictive and prognostic biomarkers into practice. So this has kind of spawned a big revolution, I guess, in utilizing omic data to try to investigate this disease. And so the Cancer Genome Atlas, I'm sure you're all familiar with, started shortly after that, and GBM was the first disease they selected. And so they had a couple of big landmark papers, Nature 2008. This is just showing kind of the summary of the major pathways that are altered in a large percentage of patients. So some receptor tyrosine kinase, basically PI3 kinase type story, NF1 with neurofibromatosis related gene. You have cell, you have cell cycle and some related to DNA damage repair and so forth. And so there have been a number of manuscripts, kind of related to this and really just a blossoming of data regarding, essentially a catalog of the omic landscape for this disease. And so the second paper that I highlight here is the Brennan paper in Cell 5 years after that initial one, and you started having once again pulling together now multi-omic data sets. And so before it was very heavy on the transcriptomic side, come up with these molecular subtypes based on the kind of the signature patterns, so proneural, neural, classical, and mesenchymal. And then now they started to basically incorporate more modern, DNA next generation sequencing, identifying particular mutations and even fusions. A big one includes the EGFRV3, which is a constitutively active tyrosine kinase that can drive the tumor, and then started pulling in methylation patterns, proteomic arrays, and things like that. So really trying to pull all this together to understand different classes of tumors. So, building all this data, now you're talking about, okay, how do we consume this? And so particularly for a non-informaticist like myself, what are ways that people have utilized this data? Well, probably the most commonly used one is cBioportal overall for TCGA, since they catalog things quite well. And have a lot of tools, continuing to update it. The Fred Hutch has this one called Oncoscape, which was first built for GBM, but actually does a lot of other diseases. Actually has some really nice informatics tools and at least some shareability of the code as well. And then there's even some more like individually controlled ones like GlioViz, which is one that is specific for GBM and other glioma data sets. So another one that I'm particularly fond of is the Ivy Gap. So this is actually kind of an academic philanthropy partnership. And so we have been part of the Ivy Consortium in the past, and this was one of their efforts that basically started to take these tumors from patients, try to follow them prospectively, a little bit more clinical annotation than we have in the TCGA, and then start getting more spatial information. So these tumors basically were histologically characterized. And so you have intratumoral heterogeneity that could be revealed. You have these anatomic structures, you have some in situ hybridization and staining, but then you add in basically some spatial elements on the transcriptome and so forth, where you have, let's say, a leading edge of the tumor or the, hyper, plastic portion, pseudo-palisade necrosis, all these different regions, and starting to get some spatial information. They have a number of research tools to go along with it. In general, though, a lot of those are more static. So you have basically, initial biopsy, and they classify it, but then you don't know a whole lot going forward. One of the things I think is evident, particularly for this disease, is that the initial tumor that you have is not necessarily what kills you, it's what survives your therapy. And so it became this interest in trying to understand really what happens after you, poke it with the stick, when you add the surgery, the radiation, the chemotherapy, and so forth. And so kind of a real world approach and collaboration was the GLASS consortium, glioma longitudinal analysis. And so the idea here was to look at longitudinal information from a number of sites. So they actually had a pretty large number of specimens for this disease. And they have a lot of high impact papers kind of on the horizon. The first one was this Nature 2019, but I understand that there are a couple that are probably imminent for being released. This was kind of more of a preview of the group with Ken Aldape and neuro-oncology a few years ago, that basically talked about, some potential patterns that might occur. Things like, okay, we know you have intratumor heterogeneity, but really, what is the potential evolution? And there really are several options. I certainly don't have time to go through all these papers, but this Nature paper is actually kind of an important one. Instead, what I'm going to do is kind of take a small review of this, because I really like their figure here, which basically just talked about these 222 patients. So the bottom line is they had DNA sequencing with matching clinical annotation. And what was, I think, part of the importance to this was that they actually found that a lot of the driver genes tended to be retained at time of recurrence. To me, there's a little bit of a caveat, because a lot of parts of the brain can't be resected well. And so perhaps some of it is because they continue to cut out the tumor in the same general location they did the first time. But nevertheless, it seemed that a lot of the tumor drivers tend to stay in place. Other alterations often depended on kind of the background of the tumor. And so there's a metabolic gene, IDH, that can regulate a lot of the biology of lower grade versus high grade gliomas. And so based on kind of that background, how you responded to therapy could also tend to drive different private to recurrent mutations. So on the GBM side, chromosome 7 gain, 10 loss is very, very common. And some of the lower grade ones, this is really the best scenario, is this IDH mutation with 1p19q co-deletion. This is the one that can potentially transition, I think, a little easier. They start having a lot of other things, including a potential hypermutator site phenotype, where you give them a DNA repair, a DNA damaging agent like temozolomide, and they have an altered repair mechanism. They can sometimes hypermutate. And so that has some implications on therapy. Another interesting consortium I was going to highlight is this CPTech group, which is really trying to combine proteomics with other molecular omic information. And so there is a paper last year on GBM by Wang et al. Cancer Cell, which took, I think it was about 90 patients and 10 normal patients who also had brain specimens, had a lot of single cell, mass spec, a lot of proteomic information, and bringing things together, and really started to bring out some things that were kind of lost maybe in the TCGA, where we started to identify tumor versus stromal elements, relationship to the immune system, other molecular signatures, and hopefully some targets for therapeutic development. So those are some of the big ones. And so just because of time, I'm going to kind of transition to, okay, what about on the preclinical side, and how can we take some of these, clinical things that really are not testable very easily outside of, let's say, a clinical trial, and how can we maybe tease out more information and utilize that. And so on the preclinical side, before I can really get into that, one of the other challenges we have, which I'll highlight here, is historically, I think we've had problems with how good our models were. And so at least in recent years, they've gotten better, but for I think a long time, we were going down the garden path because we had an improper preclinical model that really didn't reflect what was going on in an actual patient. So if we look at the traditional way that tumor models are generated, tumor cells would be taken from the patient and grown in a dish. And essentially, you would expand cells in serum with basically nutrients, and you could expand these things. And you've heard about Henrietta Lacks and HeLa cells and so forth. But basically, you can take these cells, you can put them into immune compromised mice. In let's say a flank, which would be a heterotopic site. You can put it into the brain, in this case, an orthotopic site, and you can make a tumor. The problem with this is what's illustrated here, that the original tumor that develops in the patient is doing so under very harsh conditions. It's acidic in the brain, very low oxygen, in some cases, no oxygen, like this region here, where you get flat necrosis, no blood vessels going in, and the nutrient levels are very low. But if you look at what happens in culture, so when you develop these cell lines, you actually give them everything they want to grow. It's a buffered media, you've got lots of oxygen, nutrients. And so what happens over time is you select for the fastest growing cells. And so because of that, what you end up developing in the lab, you try to treat that and say, oh, I got a great therapy. It doesn't apply to the patient because it may not be the dominant feature for their tumor. So is there something better? And so I've shown this, I think I showed the same slide a few years ago. The avatar, the patient-derived xenograft, or PDX, is a potential way to get around some of those issues. And so several years ago, I published a review on the use of these in radiation research and other cancer research. And so the idea is that instead of giving them a selection pressure in a culture dish with lots of serum, you actually have them grow in a more natural environment, let's say the flank or in the brain. And so what happens is they tend to grow more like the original tumor, and they tend to retain the molecular features of that one. The problem with them is they're costly, requires infrastructure, takes a long time to build. And then you all are doing them in immunocompromised mice. So why do we go to all these lengths to try to utilize this? Well, this just shows you the difference between putting in one of these fast-growing cells and PDX. So this is basically an H&E stain of a brain of a mouse that gets a traditional cancer line in there, and it basically looks like a marble, very sharp edge. This is what a metastasis looks like. So a lung met to the brain. It's easy to cut out, doesn't infiltrate the brain. PDX, however, tends to infiltrate into the brain parenchyma, meaning you're never going to cut it all out, and it's going to have some other properties besides just growth. We have a collaborator that used to be at UAB and now at Emory, who looked at statistical equivalence among the transcriptome for these model systems. And so we found that although in red, all these statistically equivalent genes between GBM cell lines and GBM primary tumors were very good, there's a skewing of some genes that are really elevated only in the cell lines. Most of these are growth factor or cell cycle type genes. And so no wonder when we treat some of these things, it has no impact when we do a clinical trial with it. However, when you use, let's say, a flank PDX or intracranial PDX, it gets better and better. And so we think PDXs are a nice strategy. What I'll touch on right here, really because there's no great place to put it, but we can even do additional molecular testing beyond just, let's say, the transcriptome. And so some of you have seen me talk about the Kinomics approach, but basically what we have in our kinome core is an array of immobilized phosphorylatable peptides. And so when we get tumor lysates from patients, from model systems, and so forth, we can actually do an in vitro kinase assay, in multiplex form. So basically, we can see the enzymatic activity of our sample because they will phosphorylate the different substrates on the chip, and we can get kinetic information because we pump the material through multiple times. And so we get both kinetic and an N level readout. And so we've utilized this for many people. We do, kind of clustering. This is showing an example of our initial 27 PDX for GBM, and we were able to kind of come out with, maybe 1 to 3 or 1 to 4 groups, and they didn't completely match up with transcriptomic signatures, which is indicated in these color boxes on the bottom. So there may be some convergence, maybe some divergence that are not captured at transcriptome level. So the other nice thing about this approach is because it's an enzymatic assay, we can actually spike in inhibitors and even interrogate an otherwise static sample. So we've utilized that with our model systems. I guess the main challenge with using PDX and all these things is that they're slow and they're really not built for high throughput type testings. And so a lot of our effort has also been in trying to miniaturize our models, but in a useful way. So a few years ago, we described a microtumor development program that we had partnered with a spinoff from pathology called Vivo Biosciences, which has since been basically acquired by LifeNet Health, a large nonprofit in Virginia. But essentially a humanized, a really human matrix material is utilized to embed these PDX cells, and it tends to not differentiate them and retain the original features of what they've been derived from. And so we basically found that kinomically they tended to maintain their diversity. They didn't all become the same things, at least on the kinome. When we did this, they tend to have different behaviors. Some of them tended to be very diffusely infiltrating throughout the construct. This is about a 2 millimeter construct with the green showing the live cells. It's calcium AM. And then others showed more kind of nodular formation. Some had contractile forces. They were, this is a mesenchymal one that actually can contract the construct, retain cancer stem cell fixture. So we thought it was a pretty nice model, and we've used it to try to look at as a testing bed for treating with drugs. In addition, what we've done is try to incorporate gene signatures at the transcriptome level. And so we decided a while back to utilize a custom nanostring chip. And so a former student of mine, Christian Stackhouse, now a postdoc at Duke, he helped develop basically using a machine learning approach and taking a lot of known signatures from the literature and basically whittling them down with some dimensional reduction to fit onto our chip. And then we utilize that to really profile the different ways that we culture our tumors. So not just as PDX cells or as microtumors, but even as self-aggregating spheroids, which are really another way that people are utilizing these preclinical models. What was interesting to us is that we saw that a set of genes seemed to be common to all of the ways we cultured them. And so we thought that these were really more dependent on the tumor background, the patient derived model that started it all. But if you started to grow them as spheroids or as microtumors, you started to have some genes that really seemed to be model specific, and then we had some that really didn't overlap at all. And so we thought that this type of change that was happening in culture could influence our interpretability in relation to patients. And so what we found is that if you look at the Pearson correlation, some of our tumors, like these PDX lines, it honestly didn't matter how you grew them. They retained very high correlations basically across the different culture conditions. But some, like for instance, even this X1516, if you started putting this into microtumors or spheroids, they really started to change. And so because of that, it raises concern that if we just pick one of these model systems to utilize, is this really going to be, valid? Is there going to be fidelity? And so, we started to look at some of the model independent genes. and looked at how the PDX ID versus the models played out. We looked at more model specific, where we saw, okay, all the PDX cells grouped together, and then you had the in vitro models that seem to separate, and so we see those genes that seem to do that. And then interestingly, what we also looked at were drug screens, and so we put a number of potential therapeutics on these models and looked at how they responded as microtumors, as spheroids, and some even as patient-derived xenografts in animals. And we've tried to find the genes that associated with that, basically associated with a response, both good or bad. And we saw some interesting things where this gene seemed to be increased in axitinib responding tumors, but had the opposite in erlotinib. Okay, so two different kinase inhibitors. What was, I think, most striking to us, though, is that when we try to see what genes really tended to show concordance among the models, there were a set of genes that really needed to kind of travel together when you put these in the different derivative growing conditions, and a lot of them seem to be related to the tumor microenvironment. And so that led to really the premise for my major funding right now, which is from the NIH's U01 Consortium on Patient-Derived Models of Cancer. The overall goal of this consortium was that for us to really understand how to utilize these preclinical models, particularly patient-derived models, we needed to know what happens to the biology when we use different growing conditions, different environments, and then apply perturbagens like a cancer therapy and see what happens. And so the premise that we had is illustrated here, where we kind of have symbols to represent different individual tumors from patients, and then the different representations of those. Let's say specimen from the original patient, intracranial PDX, PDX in the flank, neurospheres, which are self-aggregating, or microtumors. And what we thought was that genomically, they likely would be highly comparable, that the individual tumors, yeah, you take the specimen out, put in something else, the genomics probably look very, very similar. But when you start to get the functional omics, like the transcriptome, the proteome, the kinome, and so forth, they will start to diverge. And so one of the thoughts was, well, if we can understand what are the key tumor microenvironmental influences and modify that in the derivative systems, can we push it more towards the real thing? And so that led to this, where we think really there's intrinsic properties of the models, extrinsic pressures from how we develop those that really define how our PDMCs are going to behave. And so we looked at basically major things like chemotherapy and radiation response, and then stemness, and then incorporated a computational biology aim to try to make sense of this treatment sensitivity. And so this is just kind of showing the three aims. And so the one that's heavy on the informatics side is down here. And so Jake Chen is leading this effort, and I certainly don't have time to cover a lot of things. You probably have seen some of his presentations or his group's presentations in the past. What I will tell you about, since we don't have a ton of time left, is some of our efforts related to therapeutic resistance. And so we were fortunate enough to get a revision to this U01, which is essentially like a 3-year R01, to really delve into the tumor microenvironmental influences related to acquired and inherent therapy resistance. And so it incorporated now a 3D bioprinted system. We have a humanized mouse model utilizing a microbiome-based transplants that Brandon McFarlane is leading. And we're basically looking at more complex 3D printed systems, drug testing, molecular influences to see if we can start to identify targets. So we have 2 aims for that. So what I'm going to do really for the rest of the time is kind of talk about some of our efforts in this aspect, and then at the very end, kind of really our next steps. So all of this really began with, once again, my prior grad student, Christian Stackhouse, who kind of did a brute force in vivo study to try to determine the inherent therapeutic sensitivity of our PDX models and then develop acquired resistance models in vivo. So to basically explain it, what we have is a series of GBM PDXs. So there's like 20 that we started with. And what we did is we followed an old Mayo Clinic, which is a well-known in the PDX world for their PDX program. They're a lead member of the PDXNet consortium. And we basically put the tumors into the animal flank and then treated them with clinically relevant radiation, 2 gray, which is what we give our patients every day, every other day, basically for 6 treatments over 2 weeks. And then allow the tumors to either respond and then regrow and basically determine when they would double in volume. So it was basically a doubling time, tumor doubling time. And the time it took for these established tumors to double is indicated here. So some of them were very aggressive. This tumor took 4 days to double in volume. So very resistant after that radiation course. But some were very slow growers where you basically had 3 months before they actually doubled in size. Well, what he did is he took these ones and he started to treat them with the same thing and then passage it into a new mouse, repeat again. And so over 6 to 8 passages, what we found was that the doubling time reduced. And so we're able to select, we call it RTS, radiation selected models basically came about. And so we did this and found 8 lines that did this predictably and stably. And so what we see is that if you take the whole cohort, the median doubling time was about 35 days for the parent and 5 days for the resistant line. And so these -RT lines became a valuable resource. We believe they're the largest in existence. So we decided to start looking at these and try to see if there were things that were going to come to fruition on the omics side that would tell us a little bit about mechanism. And so overall, we didn't see really a whole lot of difference, which is consistent with like the GLASS consortium. We see a lot of the same alterations, but on pairwise specific things we sometimes did. So this JX14P, for instance, and that's the parent, and then its radiation resistant counterpart. We did see that we were seeing, some changes here that are evident. Once again, this is a different line, 1153RT with a new alteration. And when we start to look at it, we have this 12Q14 amplification. And it turns out that this actually has been seen in patients that have glioblastoma recurrence. And so our PDX model at least seems to represent what can happen in patients. In this case, you have some genes in this region that basically could contribute to radiation resistance. Is this something we could potentially target? So we're kind of excited by that. We looked transcriptomically, and similar to the DNA level, we see that in general, the radiation resistant ones don't completely separate from the parents. In fact, they tend to cluster very closely with their parent, which is not surprising based on the number of probes here. And so, yes, there were maybe some things that were globally downregulated or upregulated in the radiation resistant ones, but there really were no big slam dunks there. So on individual basis, that's where we started to see some action. So Erwin Van Meer and Satoru Asuka here basically had a model where they were actually developing radiation resistance in vivo using mouse glioma stem cells, and found similar patterns to a couple of our lines, particularly our JX39 and JX14, that seemed to be related to this N-cadherin. And it turns out that when you started to target this N-cadherin by knocking it down, you could rescue the radiation sensitivity of these models. And so once again, these systems could be potentially useful for designing, let's say, clinical trials in the right scenario. Man, I'm really having trouble advancing slides here. So what I'm really going to spend the rest of the time is kind of going over some of the informatics approaches that we've taken kind of as a group. And so I'm having trouble here. And so the idea here is utilizing a number of, golly, I'm really struggling, using a number of informatics tools to try to understand these multi-omics, particularly related to RNA interactions, network mapping, and RNA DNA. I cannot cover everything for sure. I'll spend a little bit more time on some of the things related to long non-coding RNAs, enzymatic targets, and on the kinome side, and potential small molecules. So one thing that that Christian had worked on was trying to come up with a pipeline to look at long non-coding RNAs. And so that was an interest to him. And frankly, a large number of the differential transcripts that came up in our analysis actually were long non-coding, many of which had not even been described. And so what he did is he used tools to basically pull out significant link RNAs, and then applied this thing called triple excitor, which looks for the potential for triple helix formation, which would suggest, basically regulatory properties for that link RNA, and then mapped it to the genome with bed tools, and then kind of looked upstream and downstream for potential targets. And then he took that group and then he combined with the significantly differentially expressed genes, and came up with this correlation group. And so if I can get this to advance, skipped over, we identified a number of these things. So Laura Einoff contributed a lot to this part of it, really helping try to understand some of the relationships between these long link RNAs and other groups, the RNA and DNA side. We decided to take a more targeted approach by saying, okay, we're giving radiation to select for these. Should we look at DNA repair, or DNA damage response, or DDR pathways? And so what you're seeing here is that we decided to try to combine the gene signatures for different types of DNA damage response, and then look at long non-coding and other genes that seem to correlate highly. And then this plot here, red indicates it's increased in the radiation resistant, or blue decreased in the radiation resistant. We found that different PDX tended to have different responses in the signatures that came up. So these are a lot of double strand break repair pathways that are elevated in the 39P radiation resistant ones, whereas not so much in 1153 radiation resistance, where we see, maybe more on the single strand break repairs. And then there are, a number of potential long non-coding transcripts that may influence these pathways. What we then did is we decided to combine that with some of our Kinomics things and see if we could come up with interesting networks. So this is a Metacore analysis, just showing basically if we take Kinomic information and this transcriptomic information, can we find, let's say, specific targets on a pairwise basis? So this is just showing an example from 1153 RT pairs. Here's another example. This one here, 39 PRT, really not as large of a combined group, but maybe more of a hub. Here you have Sark family kinase as a very druggable target. If we just look at the Kinomics themselves, we actually see there may be some global things. And so this PCA here shows that the radiation therapy selected, these acquired radiation resistant ones, do tend to separate from the parent at the kinome level. But once again, the pairwise comparisons really seem to be the most dramatic and really driving this. And so what we did is we took 3 of these, the JX14 PRT, the temozolomide resistant version of that, where we have the parent that's radiation sensitive and radiation resistant, and then 39 PRT pairs, and identified a number of targetable kinases in the acquired radiation resistance, And turns out that we have drugs that cross the blood-brain barrier that seem to work especially well in these models. And so right now what we're doing is we're really trying to continue to develop our 3D patient-derived models as part of our UO1, and then apply it to even more dramatic high throughput drug testing in combination with radiation, to see if we can find maybe even synergistic combos. And so we're spending a lot of time developing that. We're working on adding in normal, quote unquote, normal cell types into the model, let's say normal human astrocytes, macrophages, vascular endothelial cells, and see if we can tease out differential response to try to improve potential tolerance, let's say. And so we do a lot of that with 3D bioprinting, and applying physiological microenvironments, let's say like hypoxia, low nutrients that the patient sees. So to kind of bring this all together, I'm going to highlight another one of Jake's group's efforts in this regard. And so the transcriptomic information that I just showed you has actually been applied with this statistical enrichment analysis of samples, or the SEAS approach that we recently published in Frontiers in Big Data. So once again, I'm able to see patients that have a particular molecular profile and potential recurrence, but those patients for the most part have probably passed away and really not going to be able to help them in any fashion. On the other side, I've got these patient-derived models that look a lot like patients, but they're still tumors, outside of the patient. So how do we connect that together? And this has always been the conundrum. If you look at, how we handle these patients clinically, these patients are getting, strata, foundation, Caris type, genomic profiling. They get MGMT promoter methylation status. So we're getting a lot of molecular information, particularly on the DNA side, but how do you connect that to my models? And so maybe we'll be lucky and we'll find some key signatures, with our whole exome sequencing we've done, but is there a better way? And so this is really what they decided to tackle. And so what they did is they started plotting basically, a gene set that would basically show the different TCGA patients, and then we could map our PDX to a set of nearest neighbors. And we think that in there could be some similar biology. And so what we found was that, let's say our parent PDX that's radiation sensitive had this set of neighbors, but when we drove it to radiation resistance, it moved to a different neighborhood. And so can we use that to maybe say, okay, a patient gets a therapy, they move to a different location. Can we use information from their nearest neighbors to inform what to do next? Okay. So, so with that, I'm going to kind of conclude. GBM is certainly a devastating disease. We got to do better. The multi-omics of GBM have really just exploded in recent years, as are the complexity and elegance of the preclinical models. We do feel that PDX are very, very good models, certainly over immortalized cell lines. And so there really has been a shift to utilize these. And we think it's a potentially useful tool for looking at, really what causes demise of patients, which is acquired therapeutic resistance. We think that multi-omic information is very interesting, and sometimes may be able to globally separate, let's say, kinome level, but really it's the individual pairs, that kind of longitudinal assessment that is key. We think that long non-coding RNAs may be potential master regulators, and we're doing work now to try to validate these. But I think in terms of nearest to clinic, really the kinomic profiling, because kinases are inherently targetable with drugs, might be our kind of quickest route to making a difference in designing rational clinical trials. And so I think ultimately what we're really partnering with the informatics folks are how do we connect these models with clinical information and be able to visualize and utilize these big data sets. So with that whirlwind, I'll just kind of show a lot of the folks that are involved in this work, and there are tons, and please forgive me if I neglected to put some of you on here, but you know, people I really want to highlight are certainly Josh, who runs the Kinomics core for me. He's really probably the expert for North America on this platform. Christian Stackhouse, who did a lot of the work with our models. You have a lot of my current members. Kevin Lee is a resident fellow in my group who's been who has computational biology PhD and is really trying to help on that. Kat Langford and Yancy Gillespie's group for making a lot of these models. Laura Einov for her assistance. Certainly my other PIs on the UO1 like Anita Gemelin and Jake Chen. You know, some newer collaborators like Erwin Satoru and Ryan have been instrumental. Certainly all the folks in Jake's lab have been really, really helpful and really helping us with a lot of ongoing work. And then, you know, our partnership with LifeNet Health and others, and, you know, number of funding mechanisms listed here. And I tried to throw as many icons as I could on the right side of this. And I think with that, I'm going to stop and take any questions if there are any.
Thanks for that great presentation, and really shows here that what a collaborative effort it is. There was one chat from way back when. Is there a method to calibrate the cell line results to the PDX results? I don't know if that's, maybe that's been addressed, but that was one question that came in.
Yeah, so I think that's a reasonable one. So if you think about cell lines, you know, a lot of them really do have, you know, a big difference because you're basically, you know, growing them, they're basically bred to grow fast. There are ways to grow cell lines, certain cell lines, you know, in low adherence plates to put them in serum free conditions, which is how we handle all of our patient derived xenografts. We use stem cell culture media and techniques, and they may change somewhat. So there's maybe is some plasticity, and certainly single cell analysis of these, you know, quote unquote homogeneous cells will have, you know, will have some, you know, heterogeneity really. But there really is a big move to try to move away from this. And so I say that the more cell line results in terms of calibration, you know, I guess I probably would need a little bit more clarification in terms of utilizing them relative to clinical. What people do is they tend to do molecular alterations. So they'll genetically engineer those cells to look like patient alterations. You know, they're good for, you know, CRISPR screens, but people are doing them in patient-derived models now. The other advantage of certain cell lines is if they're syngeneic, meaning they can grow in immunocompetent mice, you can look at immunotherapy, which so far has not worked very well in GBM. And so there's some effort on that side. In terms of trying to benchmark that, really the whole goal of our UO1 is to try to determine how much manipulation of the environment is necessary to make the cell lines work as well for reading out, you know, these phenotypes versus the animals. And so that's really what we're trying to figure out is it crucial that you get the, you know, a really tight transcriptome or kinome to get the same, you know, sensitivity results? Or is it something lesser than that, something easier than that? So really for us, we're just kind of using a phenotype readout and kind of a transcriptome, kinome, you know, basically multi-ohm vector of differences or similarity really, and basing that to tell us how good a fidelity we have. And so, you know, once again, it's a spectrum, and you know, every model is you know, has caveats. So I don't know if that answers the question or not, but
to some extent, my question is because the PDX is expensive, and the cell in vitro cell lines technology is comparatively cheaper. So if we try to bridge a kind of translation, which I call calibration. So the results that we obtained in vitro is mapped to the PDX models, and we developed that model as a by calibrating. It's a calibrator, so we give an input and we give a result for PDX model. Just it's an informatics model, like a computational model, which gives us an idea that how correct is the results that we have, and then we try to calibrate it in a way that it is very close to the results of the PDX. Then we can actually use the cell line technology as PDX.
Yeah. So to kind of answer that, I still think that there are challenges with the immortalized cell lines. We've been trying that for 80 years and still having challenges. So because there are ways I can grow them as microtumors, as neurospheres, I can get them to attach by using different matrices. I mean, people have 1536 well arrays that, culture dishes that you can do these studies. And so that's what we're doing. We're more on the 96 well, kind of a semi-high throughput. And we believe that the PDX, yes, it's maybe a little bit more expensive than cell lines, but they're vastly superior. So our view is that if we have a library of PDX or neurospheres or microtumors that are at least representative of human disease, we probably have a close enough match that most patients will kind of fall into a bin for relation. And so you don't have to have just this unlimited, you don't have to make 17,000 models. Could you get away with 50, 100? We've got about 60 something here in terms of PDX. We have more on the spheroid side. That could be sufficient, perhaps. And so we think that all the things you mentioned about trying to make these informatics and computational tools are still worthwhile, but we should do it with a more relevant model. So don't grow them in bovine serum with 10 times the amount of sugar that any patient would ever have in their body, things like that. And so we think that growing them smartly, where you pay attention to the microenvironment, will give you better readout. And then you still can use all those computational approaches and do a lot of the work in an ex vivo setting, which is really what we're trying to do. So it looks like we have another question about the immunotherapy. So I moved to this slide. So in general, immunotherapy has failed for GBM. If you look at things like tumor mutational burden, glioma is actually on the lower side compared to, let's say, melanoma, maybe head and neck, and lung cancer. However, there are settings where we think it may be useful. And so I bring up this hypermutator type. So this was first described kind of in the mid 2000s, where you had these patients who were getting temozolomide for an early glioma, which usually is good, pretty decent prognosis. Patients live many, many years, that the tumor behaves. But there are these subset of patients that would come out and their tumor would come back with a vengeance with lots and lots of mutations. They call them hypermutators. And they thought it was because they had this IDH mutation. They end up having, this alkylating chemo, and it just led to just the floodgates in terms of tumor mutations, which led often to basically a high grade glioma like a GBM and bad outcomes. Well, it turns out that actually may be a good thing if you're thinking about immunotherapy. And so we're using tumor mutational burden as a potential means to give immunotherapy. So these hypermutators are probably ones where immunotherapy can work. I would say where a lot of the work related to this is more in cell therapy. So we have a clinical trial here from UAB related research on gamma delta T cells. We have oncolytic virus clinical trials going on. But I think on the immune side, I think maybe the combination of radiation, which tends to kick up the immune system some, and basically careful selection of the right patient and give immunotherapy is the right way to go. Other questions?
I have a question, and this is not unique to glioblastoma, that, it's like we would like to be able to sequence everybody. Anybody gets a cancer or a biopsy or something, it's malignant. Let's biopsy everybody before we start exposing to things that cause resistance and limit later treatment options. But that can be expensive. And so, maybe you get most people or a good percentage of people with the standard, just the standard off the shelf kind of treatment or the typical treatment. So how do we balance that, or where do you see that heading in the near term? Whether everybody gets a full sequencing and determination of all these factors, and growing in a mouse model, or do you think that people are going to be treated with a standard first, just because not everybody might need that, but then when you're talking about something so aggressive like GBM, that maybe you have a different.
Yeah, so great question. So we've kind of taken, I guess, mainly a practical and realistic approach. So if we have no great alternatives, then we're really trying to find something that works. So probably virtually all GBM patients get molecular profile, okay? And we're starting to get richer data sets. So you start off with just a few things, more, very focused. Now, you start having things like Caris, where they're starting to do like, almost really like whole transcriptome, large genome, hotspot, alterations. There are fusions that are rare but potentially targetable that we see. So we're trying to find that needle in the haystack. For other diseases where, we have a very effective standard of care and not a great set of predictors for recurrence yet, one that I treat a lot is head and neck. We don't typically do a lot of molecular profiling other than very simple prognostic markers like human papillomavirus related tumors, which have very different outcomes. We tend to reserve the omics for recurrence. And so obviously as it gets cheaper, faster, more accessible, I think we're going to add more and more to the list of tumors that we are going to molecularly profile, because I think at least there's some interest by the pharmaceutical companies and insurance companies that, hey, if you're willing to screen with a modest cost, let's say next gen sequencing, and I can let's say, oh, this patient probably won't benefit from a drug that costs $20,000 a month, it may be in their interest to profile everybody. But for others, it's really more when the standard of care is just either not satisfactory or you're kind of running out of options. And so, how academic centers incorporate the molecular tumor board has changed even just in the last several years, with the option of immune checkpoint blockade. It used to be, okay, do you have PD-L1 expression? Now we're getting the CPS, TPS scores. Now it's like, you don't even have to check that. If you have a tumor mutational burden that's high, you can get some of these therapies. So, a lot of people are like, what's the most information we can get from a test? And so, right now, I think Caris is one of the best ones. And so you're seeing that being incorporated more and more into clinical practice because it's going to potentially affect what we offer patients.
Yeah, thanks. Unfortunately, we're out of time, and this has been a really great talk. And thank you for sharing your new developments with us. And it's good to see that we're moving in this direction, especially for a advanced aggressive disease like GBM. I want to put a quick plug in that a lot of people think that, well, typically people speak in this series by invitation. And so people might think, well, I'm not worthy and whatever, but I want to just change that. And, we're always looking for new speakers, finding out what things are going on. And, if you're interested, you have something to present, you're doing some interesting work, you have trainees who you want to have them get out there, get their work out there, get them recognized. All this is very important for tenure promotion, finding jobs and opportunities and networking and everything. So So I encourage everybody to either volunteer themselves or volunteer other people, or even just let me know if they have some great speakers that they know of that would be good for the series. But anyway, so thank you very much. Thanks for attending and staying around to the bitter end. And Happy New Year to everybody. And we hope to see you at the next one. So thank you very much. Have a good weekend.